So we're going to finish out Unit 2 with the macromolecules. We've given ourselves some background information on the chemistry that is involved with biological systems, but now let's talk about some specific categories of molecules and that we find in the human body and we find in living things in large quantities or in high amounts. So why should we care? Right? These macromolecules, these different categories, are really the building blocks that make us us and make all the other living things, right? So without these building blocks, we wouldn't be able to do all of the functions that we seek to accomplish, right? Our cells wouldn't be able to carry out their different processes, and larger organisms like ourselves would certainly not be able to exist, right? There's 4 categories of macromolecules we're going to get into: carbohydrates, lipids, proteins, and nucleic acids. And each of these categories have certain features and functions and characteristics that we're going to get into. So let's go ahead and get into our first category, carbohydrates. Carbohydrates are mainly built of monosaccharides, also known as simple sugars. Mono meaning one and saccharide being sugars. And we find that there's energy that can be stored between the carbon bonds. So in carbohydrates, hence the name carbohydrates, there's a lot of carbon and a lot of hydrogen. And these carbons are linked together. So you have chains or rings of carbon. The energy that we want to store away or use later is stored in those carbon-carbon bonds. So the bonds held between 2 or more carbon molecules or carbon atoms. If we look at the category carbohydrates, we can break them down into 2 distinct groups: the simple and the complex carbs. Simple sugars or simple carbs include the monosaccharides and the disaccharides. Mono, I already mentioned, means 1. Di means 2. So these are really small carbohydrates, things that are only going to have 1 sugar or 2 sugars and nothing more. So some examples are things like glucose, which is used for fuel. We'll talk about glucose in later chapters. Fructose, which is found in fruit that you eat. And then sucrose, which is a disaccharide, which is table sugar. So this is what those look like, right? And we're not going to worry so much about the ball and stick models, but we'll use these instead. So glucose is pretty small, right? 6 carbons, that's it. All found in a ring with various hydrogens and oxygens hanging off of them. Fructose is the same makeup, just with a slightly different arrangement, but same kind of ring-like structure, just slightly different from glucose. And then sucrose, or table sugar, is 1 glucose and 1 fructose that have been linked together. But even then, not a very large molecule. This is actually not massive when we're looking at the macromolecules. The larger carbohydrates are known as polysaccharides, or many sugars. These are huge. We're talking about hundreds of things like glucose all stuck together. These are really, really, really big. Right, so here's a couple that we can look at, and these are ones, these molecules are molecules that can be used as a storage for carbohydrates. We can store energy away between those carbon bonds in this fashion. But depending on what kind of living organism you are, you're going to use a different type of polysaccharide to store it. So if you are an animal, you'll use glycogen, right? So glycogen is a really large polysaccharide that animals will use to store away carbohydrates in their muscles as well as their liver. And this is for quick bursts of energy, right? So we can use this when we are kind of like sitting around and then something happens, we got to get up and run. Maybe all of a sudden you see your cousin or brother or sister or child reaching for something they're not supposed to grab, right? And you immediately get up and you're just, I'm going to stop you from doing this. You don't hurt yourself, or you don't, make a mess. Versus if you're a plant, you use starch. This is stored in roots and stems, a really A good example of this would be things like potatoes. If you eat potatoes, it's a lot of starch. It's a polysaccharide, it's a complex carb. This is for long-term energy storage, so it's not necessarily something that's going to be used right away. And when you notice the difference between these is that the glycogen, you see how all these different molecules, and this is just a snippet, right? But you see how it branches off versus starch, which is just a long, long chain. There's a few other polysaccharides that are used that are not for energy. So glycogen and starch that we just talked about, that was energy storage. But some polysaccharides for non-human animals and for plants can be used to provide structure and support. So cellulose is one of those, and that's going to be found in plant cell walls. It's going to help kind of give some structure while also maintaining some flexibility. So this is why plants can move around, but they can also hold themselves upright. And chitin, which is found in the exoskeletons or the shells, so to speak, of arthropods. So insects, think crustaceans, arachnids, so things like crabs and shrimp. That outer kind of shell, so to speak, it's not really a shell, but that outer kind of skeleton they have is made of chitin. It's also found in fungus cell walls, and it's very rigid. So this is much more, it's much stronger and less flexible than cellulose. Both of these are indigestible, meaning you can't digest it, you can't get nutrients from this if you eat it. It doesn't mean that it's not good for you. So something like the cellulose, when you eat plant matter, actually is helpful. It's fiber, right? You can't digest fiber, but it does provide help when it comes to defecation, right? Building up some bulk so that it's not super hard and dry when you have to defecate. Moving on to our next category is the lipids. Lipids are mainly carbon and hydrogen. That's the most part. Most of the molecules are going to be made up of those two elements. They're very energy dense. So these, you can pack a lot of energy in the bonds of lipids. One really important feature that is shared among all lipids is that they are not soluble in water, meaning they do not dissolve in water, and they have a greasy or an oily kind of texture. So I just mentioned they don't dissolve in water, and there's some terminology we want to use to describe that besides just that. And it has to do with the chemical interaction of lipids and water. Now, with lipids, there's a lot of different kind of structures to lipids. We'll talk about a little bit of that. But the structures themselves don't really have much in common, right? We saw with the carbohydrates that we have like these glucose or fructose molecules, and we kind of just glue all of them together into these long chains and manipulate them that way. But with lipids, there's a lot of different ways you can arrange things. So how do we categorize them all together? What unites all of them together? What unites them is their insolubility, their hydrophobic nature. They don't dissolve in water. We call that hydrophobic, also known as water insoluble, also known as nonpolar. So if you remember when we were talking about the properties of water, water had those partial charges, right? And this is why it could form hydrogen bonds so easily, is that oxygen had a slightly negative charge and the hydrogens had a slightly positive charge. In lipids, you don't have that. Everything is equal. There's an equal distribution or even distribution of the electrons versus something like water or things that are hydrophilic that also like water or are polar like water. Those electrons are not evenly distributed, and that's what gives that the uneven charge, right? Those partial charges. So if you dissolve something or you dump something that is polar into water, it will dissolve because they share that property of the uneven electron distribution. But if you pour oil into water, oil has a very even distribution of electrons and water doesn't. They don't share that property. They cannot mix together. right? And you can see this if you go to your fridge. Go to your fridge. If you have something like an Italian dressing or some sort of like oil and vinegar or oil and water mixture, like a salad dressing, look at the bottle in the door. You've noticed that a lot of things have settled, right? You give it a shake before you put it on your salad because otherwise you won't get all the dressing. You give it a shake, you mix things up, but if you set it back down, things start to settle and separate out again, right? And so every time you want to go use that dressing, you have to shake it up again because of this issue, the hydrophobic nonpolar molecules that don't want to interact with the hydrophilic and polar molecules. That being said, we do need fats and oils in our diet. It's one of those major categories, and we use them throughout our body. We can get them from food we eat on a fairly regular basis. And in general, when we're talking about dietary fats, we're talking about triglycerides. Triglycerides are called so because we have a glycerol backbone, and then we have three, hence the name three fatty acid tails coming off of that glycerol backbone. Now, how we arrange those fatty acid tails and the bonds we have between the carbons will determine if we're saturated, unsaturated, or trans fats. Saturated fats are called saturated because they have single bonds between all of the carbons. And so you've saturated the carbon tails, the fatty acid tails, with as many hydrogen atoms as possible, right? They have the maximum number of hydrogen bonds, or hydrogens bonded to the carbons. And so because of that, it's a solid at room temperature. Those tails are very nice and straight, and we can pack them together. These are really delicious. Think of like your butters, fat on a nice steak. These tend to be things that are animal in nature, but they do contribute to heart issues. Unsaturated fats are the oils. So think of like olive oil or vegetable oil. These are primarily a liquid at room temperature. These are called unsaturated because some of the carbons, one or more of the carbons in those tails, have a double bond. That double bond puts a kink in the tail because we can't put as many hydrogens on those carbons, and so you can't tightly pack together. There's going to be space between the fatty acid tails and between the triglyceride molecules. So since we can't pack together, we end up being a liquid at a room temperature. These are healthier for you, right? There's unsaturated, and then there's also polyunsaturated, which just means that you have a lot of those double bonds. And then lastly, there's trans fats, which are not really found in nature. These are not good for you. These are fats that you should avoid all the time. You should not consume them. Saturated and unsaturated, you can have some saturated fats in your diet, that's fine. Unsaturated is definitely the healthy way to go, but trans fats, definitely avoid them. Your body doesn't really exactly know how to process them very well. Some other categories of lipids include the sterols. So these are things that are steroids, they're lipids, but not fats. So all fats are lipids, but not all lipids are fats. And this is why I said the lipid category is kind of broad and very diverse, and that the one thing that unifies them all is them not mixing with water. So the sterols, like cholesterol, and the sex hormones like testosterone, estrogen, you'll kind of see it in the name. You'll see the sterol or sterone or estro, something like that in the name that gives you that hint. Their structure is basically 4 carbon rings that have been fused together, and then various groups that are added on to that. Cholesterol is going to help with the membrane of your cells, but cholesterol also is bad if it's in excess because it can increase your risk of heart attack and stroke. And then sex hormones, you need those to regulate your growth and development of the sex organs. Phospholipids is another category. Phospholipids are pretty special. These have a hydrophilic and a hydrophobic part to them. So we've replaced part of the triglyceride, we've taken a fatty acid tail off, and instead we've added a phosphate group to the glycerol and backbone. So with the phospholipids, we end up with one side that's hydrophilic and the other side that's hydrophobic. This is what's going to make up your cell membranes for the most part, and it's that dual nature of the phospholipid of one part hydrophilic, one part hydrophobic that allows it to be that membrane that can help control what comes in and what goes out of the cell. We'll talk more about that in a later chapter. Right, so here's just some examples that we see. We see the cholesterol, we see the sex hormones, and we see the phospholipid. Right? And so if we look at the pictures, we see that 4 fused carbon ring backbone of the sterols. And then we see that on our phospholipid, we've removed one of the fatty acid tails, and instead we have this different looking head. Moving on to our third category, we have the proteins. The proteins are a very diverse, very wide-ranging group of macromolecules that do so many different things in our bodies and in living things. Their subunit is called the amino acid. This is the basic unit of proteins, and when we link a lot of amino acids together, that's how we build up the proteins. We link them by peptide bonds between carbon and nitrogen. Sometimes proteins are called polypeptides because of this. So anytime you see the word peptide, you should think something to do with a protein. It's either a piece of a protein, the bonds found in a protein, or if it's like polypeptide, the protein itself. And like I said, proteins are really diverse in what they can do. We use them to help us grow, we use them to repair ourselves, to store away energy, to transport things, to signal other things. You name it, and there's probably a protein that does a similar function. But the diversity of all these different proteins only comes from 20 different amino acids. It's the specific arrangement of those different amino acids that gives us our very special, very unique protein. Now, with the amino acids, we can make 10 of them. So half of them we can make ourselves, and these are considered non-essential. But the other 10 we get from food. We cannot make them ourselves. So these are called essential amino acids. You have to consume them from the food you eat. Not all of these can be found in every single food you eat. Some foods are complete. They have all of these 10 essential amino acids. Others don't. Usually, the complete foods that have all of these are animal products, although some plant products do have them. If you're something like vegetarian or vegan and you don't consume animal products, that's okay. You just have to have the right combination of foods to make sure you're getting all 10 of these essential amino acids. Now, we mentioned there's only 20 amino acids, and yet these proteins, there's tons of them. They all do different things. How the heck do they do that? How do they get so different and so unique based on only 20 different building blocks? Like I said, the arrangement of them is important. The function of a protein is determined by the shape it takes on. So let's look at protein shape and how that's determined. How do we get the unique protein that does this particular function that makes it different from all the different proteins out there? We start with the primary level of protein structure. Primary level is kind of like just the order. If you were to take a protein and take all of those amino acids in that protein and string them out in a long line like a string of beads, that would be primary sequence, right? And we see that here. We see each of these amino acids just all shown out in one single line connected by their peptide bonds. Then we take that long string and we start adding in some localized folding. So this is our secondary level of protein structure, where we're adding in some localized folding, some localized structure between the amino acids. So things like an alpha helix, shown here, or a beta sheet, which is like a pleated look. Then we get to our third level, the tertiary level of protein structure. This is a 3D shape of the protein. Now, some proteins stop here, right? If it's only a single string of amino acids, one long polypeptide, this is the end of the road, right? So this is a whole 3D shape. all the sequence, all in order, the secondary structures, and then how these secondary structures are formed around each other. Now, if our protein has more than one polypeptide chain, or more than one string of a polypeptide, we get to the quaternary level of protein structure where other proteins can associate with it. And so how the other proteins associate with it, like how they arrange themselves together, is quaternary level of protein structure. This protein structure is super important though. If we don't have the right shape, we can't actually function. We call that denaturation. So when a protein is denatured, we lose the 3D structure. We lose this structure here. So the order, the primary sequence is still there. We still have the correct order of amino acids. We haven't broken everything down. We've just changed the shape. And that can happen due to various things. It could happen because we've heated something up or cooled something down. Maybe we've changed the pH. Maybe solutes have changed. Basically, something in the environment has changed drastically enough that the protein can no longer hold the 3D shape it has. So that shape changes, or we lose the shape completely. And when it does that, we call it denaturation. The protein has denatured, and when it does that, we lose function. It's not going to work anymore. So let's say that particular protein was an enzyme, and an enzyme is a special protein that can start or speed up chemical reactions, specifically in living things. If we change the 3D shape of that enzyme, it's not going to be able to work on those chemical reactions anymore. It's not going to help us anymore. So enzymes are very specific, right? They can only work with certain things, and they can only work under certain conditions. They're kind of like little divas, if you think of them that way, and that they have to have the right working conditions, and if they don't have the conditions they want, they are not going to work. And they can only work on certain things, right? We call that a substrate. So the enzymes, if they don't have the certain substrate, they're not going to work on anything else. And if they don't have the right environment, they're not going to work at all. That takes us to our last category, the nucleic acids. Nucleic acids are made up of a subunit called the nucleotides.